Hello everyone, and welcome to this video on CRISPR-Cas9. Throughout this video, we hope that you gain a basic understanding of its history and purpose, the three basic phases of the technique, some associated applications, and current work that is being done with it, as well as current limitations. To start off, CRISPR was first discovered in the late 1980s, and was derived from the antiviral immune system of bacteria. Upon realizing this natural mechanism of cutting out unwanted segments of viral genomes and plasmids, scientists soon realized that it could be used as a gene editing technique. Currently, there are three types of CRISPR-Cas systems, but in today's video, we will be analyzing the emerging technology of the Type 2 CRISPR-Cas9 system, which was discovered in 2012 by Jennifer Doudna et al. This system can be used to edit genes in three ways. The first one is disruption, in which a single cut is made and enzymes in the cell repair the DNA through a process called non-homologous end joining. This will ultimately result in the addition or deletion of base pairs and disrupt the original DNA sequence, leading to gene inactivation. The second way is through deletion, in which the DNA is cut in two different spots, and in a similar process to disruption, the DNA is repaired to unite the separate ends and discard the sequence of interest. The last way is adding an insert, in which CRISPR-Cas9 works with a DNA template to correct or add a gene through a process called homology-directed repair. The Cas protein prevents bacterial cell infection by cleaving onto bacteriophage DNA. The DNA is then placed into interspace regions, where it can later be transcribed into RNA to guide the Cas9 nuclease to the invading bacteriophage DNA. This RNA is referred to as guide RNA, or gRNA for short. Now that we have established how the Cas9 nuclease works, we are left with one question. How can we use Cas9 for gene editing? Well, we can target certain nucleotide sequences by designing our own gRNA. This gRNA is typically a 20 nucleotide guide that pertains to the targeted gene. The gRNA identifies a particular sequence of the target gene and helps guide Cas9 to it. The Cas9 enzyme attaches itself to the DNA segment and begins to cut at the site. The site where it attaches is known as the protospacer adjacent motif, or PAM for short. The Cas9 enzyme acts as molecular scissors which cut into the DNA. This results in a double-stranded break. After the cut has been made, the cell tries to repair the DNA. The cell can undergo two different repair processes, non-homologous end joining or homology-directed repair. In non-homologous end joining, small insertions and deletions, which are referred to as indel mutations, can occur. Non-homologous end joining can also be used to substitute certain nucleotides. It is useful for gene knockouts, but it is prone to errors. Homology-directed repair is another process. A DNA repair template containing the desired sequence is put into the cell with gRNA and Cas9. The sequence must have edits and homologous sequences that are further up and downstream. It can consist of single-stranded oligonucleotides, double-stranded oligonucleotides, or double-stranded DNA plasmid. Homology-directed repair can be superior for precise gene edits. Finally, research techniques such as PCR or DNA sequencing can be used to ensure that CRISPR was successful in making genetic edits. CRISPR truly has implications in a variety of disciplines in medicine. Its ability to correct and remove genetic information to improve the quality of life of the patient speaks to the emerging field of research. Some applications of CRISPR include CRISPR's role in cancer research, providing researchers the ability to simulate and treat similar cancers to those seen in humans. CRISPR's ability to correct small genetic changes which cause life-changing disease, such as sickle cell anemia and beta thalassemia. And lastly, CRISPR's interaction with stem cells, which have the ability to pass the blood-brain barrier to treat neurological disease. CRISPR can be used to genetically modify so many things. One of those is a DNA sequence that encodes for cystic fibrosis. But first, what is cystic fibrosis and what causes it? Cystic fibrosis is a genetic disease. It is caused by a loss-of-function mutation in the gene that encodes the protein called the cystic fibrosis transmembrane conductase regulator, also known as CFTR. When functioning properly, the CFTR channels transport water and charged ions, which will then create a thin lining mucus around organs. In individual cystic fibrosis, the mutated CFTR creates a variation of the water and charged ions that instead of creating a thin mucus, it creates a thick mucus that can block the airway and can confine bacteria. Here on the left is a diagram that describes how the water and chloride ions normally function within the CFTR channel versus how they function in someone with cystic fibrosis. In the diagram, you can see that there's a buildup of mucus in an individual with cystic fibrosis. By using gene editing, CRISPR can help to stop the buildup of thick mucus. CRISPR can change the DNA sequences that encode for the mutated CFTR gene and can replace it with the correct version of it. How does this work? CRISPR and the enzyme Cas9 bind to a guide RNA that can recognize a mutated gene and then cuts it out. By doing this, the cell can replace the missing sequences with the correct ones by filling out what's missing. Now that we have discussed how CRISPR works and its applications in gene therapy, we would like to cover some limitations of this technique. The main limitation using CRISPR-Cas9 is off-target mutagenesis. These off-target effects produce unintended mutations at different DNA segments. New engineered Cas9 variants and optimized guide designs can help reduce the chance of off-target effects. Another limitation is the choice and use of delivery methods. Typically, adeno-associated virus vectors are used in gene therapy applications due to their non-integrating nature, compatibility, and high transduction efficiency. However, adeno-associated virus vectors have a limited carry capacity, which means it can only be used to deliver small sequences of DNA. They also may have limited tissue tropism, as they're more effective at targeting certain cell types, limiting the application of CRISPR for certain diseases. Another limitation associated with the delivery system and the use of Cas9 is the need for a protospacer adjacent motif near the target site. Cas9 nucleases can have different PAM recognition sites. Typically, Streptococcus pyogenes Cas9 is used due to its short PAM recognition site. However, due to its larger nature, it can be more difficult to package into adeno-associated virus vectors. Other Cas9 nucleases can be used, but they may have longer PAM sequences, limiting the number of genes that can be edited. DNA damage toxicity is another limitation of CRISPR. This is because double-stranded DNA breaks can trigger cell apoptosis. What's interesting is that cell apoptosis mainly occurs in cells that demonstrate p53 activation. Therefore, there may be a bias for oncogenic cell survival. Successful CRISPR attempts edits more frequently in p53 suppressed cells. Immunotoxicity is also a common limitation. Individuals can possess pre-existing immunity, especially when they had prior exposure to adeno-associated viruses. The immune system may attack and destroy the adeno-associated virus vector before they can even deliver the desired genetic material. This can also pose a safety and health concern. To conclude this video, we hope that you gained valuable insight into the processes behind CRISPR-Cas9 systems, the different applications of CRISPR, and the limitations associated with the technique. Thanks for watching.